Hi, in this lab, you are learning how to use tables and graphs to effectively present your data. In other words, to present the results of your experiments. And just as scientists have a very uniform way in which science is conducted, called the scientific method, there's also a very uniform way in which science is reported. In other words, the way that they present their results is very uniform throughout all disciplines of science. So if you look at any typical peer-reviewed scientific journal article, you should see that the entire paper really is published in a very uniform format. So just quickly looking at a scientific journal article as an example, I got this, by the way, from the Palomar Library website. As students and faculty, we have access for free to thousands of peer-reviewed scientific journal articles, which is very awesome because these would normally be very expensive to access. So this study was looking at the effects of climate change on the population ecology of polar bears. I have not read this study. I know that because this is an excellent, well-written title. The title should tell the reader exactly what was done in the study. The name of the journal is Arctic, tells the volume number, year, pages, all of that. Scientists are there. The first section is always the abstract. And in the abstract, within one paragraph, this is telling you the basics of the study, what they did, what they found out, what it means, all in one paragraph. Next is the introduction. And within the introduction, you typically see lots of references because this is where they not only explain what they're planning to do, but the science behind what they're planning to do. We got to where we are today in science through shared knowledge. And scientists are either trying to replicate what's been done before or take it to the next step. We're going to add knowledge to our current knowledge. Next is always materials and methods, telling you how this study or experiment was conducted. And then next, we have the results section. And in this results section is where we should expect to see graphs that tell the story of the results and data tables. So I'm mostly interested in the graphs. And yay, here are some graphs. These are called scatter plots. And scatter plots are excellent for showing the correlation or the relationship between your dependent and independent variables in your experiment. And in these scatter plots, the x-axis, which is this one, always shows your independent variable, whereas the y-axis, this one, shows your dependent variable. Remember that the independent variable is the thing you're manipulating or changing, and the dependent variable is dependent upon the independent. So in this case, whatever breakup is, I'm assuming it's breakup of ice, is dependent on what year it was. And then the scientists have very appropriately inserted a trend line so that it's not just a bunch of scattered data points. They're showing the actual trend in the data. So scatter plots will be used a lot in this course. They are used to show the relationship between your two variables, your dependent and independent variable. This is called a line graph, and line graphs are very appropriate for showing change over time. So you can see years on the x-axis, and you can see this change over time with the line graph. Bar graphs are the third category of graph we're going to look at today. I will show you an example of that when we graph in Excel, but those are not as frequently used in this course. They're only used in very specific circumstances. If we had a bunch of bars here, it would be very hard to see this trend. Okay, so now I need to stop sharing this document and open up this document. Sorry. Do a new share. Okay, so this is a typical data table, and this is actually from an experiment from this course, looking at the effects of enzyme concentration on reaction rate. And you can see this is the average. So in other words, there were multiple trials of this experiment. And at the end, we're going to graph just the average. So to get to this average, we had to average a bunch of data in a separate table. And now I've set up this table this way because this is the data I'm going to graph. Now, if we were putting this into a scientific paper, we would not only want to call this table 1 or whatever the number is, so that we could refer to it in the text of the document. We could refer back to this as table 1, but we also want a title. So I'm going to call this effects of enzyme concentration on reaction rate. Your title should always tell you exactly what was done. It shouldn't just be table or graph. Now this is looking at the effects of one variable on another variable. And whenever you're looking at the effects of blank on blank, this is the thing that's being altered or changed. That is your independent variable. So enzyme concentration is our independent. We should see that on our x-axis. Reaction rate is our dependent. We're saying reaction rate is dependent upon the enzyme concentration, and that should be our y-axis. The cool thing about Excel is there are multiple ways to do almost everything in Excel. I'm going to show you the way that I like to graph in Excel, but I realize there are other ways to do it. We want this to be a scatter plot because we are looking at the relationship between two variables. This isn't change over time. This isn't categories of data where we would use a bar chart. So I'm going to click in any cell out here, and I want to insert a scatter plot. So I'm going to come up here, and I'm going to click on insert. Now this gives me a lot of options. I want to insert a chart. And I want to insert a scatter. Okay, so see, it didn't do anything because I haven't told it what to graph and in what format to make the graph. Okay, so for quick layout, I'm going to choose the graph that has everything because I want a title. I want X and Y axis labels with units. And sometimes I'm going to need this legend box. Now today I don't need it for this graph because I only have one data set, but I'll show you an example of when that legend comes in handy. But I just automatically pick the one that has everything. Now I want to tell it what to graph. So I'd go up here to select data. Okay, so I said insert scatter plot. I went to quick layout to tell it I want it to have every possible piece of information. And now I'm going to tell it what to graph. So I'm going to hit select data, and I'm going to highlight just my numbers. Say, okay. Awesome. So here is my basic graph. Now I need to do a few things to it. I'm going to label my x-axis, these percentages, that enzyme concentration, and in particular, it's catalase enzyme. So I'm going to call this catalase concentration. It's percent, but percentages are already on the numbers. If those were just numbers, I would have to put a percent as my units. All well-constructed graphs will have axis titles with the appropriate units if necessary. This is our reaction rate, our y-axis, our dependent variable. Okay, and that's centimeters per second are our units, so I'm going to put those in parentheses. Now we need a title. Effects of catalase enzyme concentration on reaction rate. So your title always needs to tell exactly what's being shown in this graph. You would never say graph 1 or figure 1. I don't need a legend, so I'm going to delete that. But what I do want in this graph is I want to see the trend in my data. Now I can tell that it's increasing, but I want to insert a trend line. And I'm going to show you a couple of cool things about that trend line. So to get the trend line, I'm just going to click on any of these data points. You can click on this one, you can click on any of these. And I get an X on the data points. Now I'm going to right click and say add trend line. And once I do that, I have this nice trend line, so I can now see yes. As catalase concentration increases, reaction rate increases. Over here, I now have a menu to do a couple of things with that trend line. I want to set the intercept to 0, meaning now my line goes all the way to the 0, and I'll show you something cool that now allows me to do. And I also want to display the equation on the chart. Now, this trend line will allow me to predict what the reaction rate would be at other catalase concentrations. So I can say, hey, at 90%, so 90% would be right here. And if I go up to my line, I can go across and see that the reaction rate would probably be exactly 0.1. The other way I can determine that is I can use the equation. Okay, so Y is my reaction rate. So in other words, reaction rate is equal to 0.1167 times my X, which is catalase concentration. So you can plug the catalase concentration into this equation and predict the reaction rate exactly using that. So that's a scatter plot. I want to just quickly show you other graphs, and then I'm going to show you how to really effectively copy and paste these into a lab report in Word. Okay, so this data is an example of a scatter plot where you would not want a trend line, because this is showing the relationship between temperature and enzyme activity, and it's not just increasing. You can see that the numbers are they kind of seem all over the place, but let's see if there is a trend here. So we're going to choose a scatter plot again, because we're not looking at change over time. We're looking at the relationship between two variables, temperature and enzyme activity. That is best shown by a scatter plot. So I'm going to insert, chart, scatter, quick layout. I want everything. Select data. Numbers. Okay, now I want to make sure that this is temperature. Yes, it is. And then this is my enzyme activity. So I would complete those axis titles, and I would put a title on my chart. Again, I don't need this because I only have one set of data. Now, this is a unique situation where it's going up and then it goes down. Well, I'm not going to say it's unique, but it's different from the previous one. What this is showing is the highest enzyme activity was at 35 degrees. Anything below 35 degrees Celsius, the enzyme activity went down, and anything above 35 degrees Celsius, the enzyme activity went down. And you'll learn in this course why that happens. So this is just an example of a scatter plot where you wouldn't do a trend line. The data points kind of just speak for themselves. You wouldn't do just a straight line with a formula. Okay, now let's look at an appropriate time to use a bar graph. Okay, so this is categories of data. We have these age categories. We also have two other categories, men and women. So this is very appropriately graphed in a bar graph, also called a bar chart. The other thing you could do, and I don't always recommend doing this because it seems to always want to do a bar chart, which is not always the best. Okay, but you could also just highlight these numbers and you could say insert and then recommend charts. And yay, it tells you bar chart. The other thing you could do though is you could just say, I'm going to do a bar chart because I know that's the most appropriate for this data. So you would say insert, bar chart. Again, we need to do our layout. that has everything labeled. So I just always pick the one that has the most stuff. So layout 9 on this. And now I'll select my data. And highlight all my data. And ta-da! Again, I would change my axis titles. I would give this a title that said this is average cholesterol level by age and gender. We can see the trend. We can see that men tend to trend a little bit higher than women. And we can also see that total cholesterol level tends to increase with age. So this is an appropriate use of a bar chart or a bar graph. Okay, now last would be our line graph. And again, that would be looking at change over time. So this is looking at the growth of E. coli bacteria by day. And I'm actually going to change this to different numbers just because I want to show you something that I can't really effectively show. The line graph is going to be done a little differently. So insert, there's my icon for my line graph. I'll just do one without data points for now. Layout. I want one that has titles. We might still have to add another title. And now I want to select the data. Now this is where it gets a little different. Chart data range, that is just your Y data. And we want our Y-axis to be the number of E. coli colonies. So I'm just going to highlight those numbers. Now for the horizontal, the X-axis, I want it to be the days. But what this thinks I want it to be is data point 1, data point 2, data point 3. So I have to tell it that's not what I want. I want to edit my horizontal axis. And now it's saying, well, what do you want it to be? What I want it to be is 2, 4, 6, 8, 10, 12, 14. Ta-da, 2, 4, 6, 8, 10, 12, 14. Now my number of E. coli colonies, I can clearly see that trend increasing over time. I mean, if you wanted, you could change this to E. coli bacteria colonies on the chart title there. I'm sorry, on the legend title right there. Now I would want to insert an X-axis title. It's not really letting me do that. I would have to choose a different, I think I chose the wrong quick layout. So you would want to choose the layout that already had that there. For some reason, it's not letting me add it. Okay, but that's a line graph. That's change over time. Bar graph, category data. Looking at the relationship between X and Y, in other words, dependent and independent variable, that's going to be a scatter plot. Now I just want to quickly show you how to copy and paste these into a document. By the way, I put these grid lines on here purposely so when I copy this table from Excel into Word, it will look nicer. I want to just quickly show you something, how I did this. I'm going to undo this one. Get rid of those borders. I'm going to okay. So say your data looks like you just typed this in and here it is, but now I want to copy and paste this into my lab report and I want it to look nice. I want some grid lines around my cells, and to do that I'm going to go right here and I'm going to tell it I want all borders. Okay, now this looks pretty sloppy the way it is now. It's going outside of the cell. So now I want to take this and I want to right click and say format cells, wrap the text. And now it's ready to just copy and paste into another document. So let's just quickly do that. I'm going to copy just my table. And you can either right click and tell it to copy or you can just say Control C. And when you see this dotted line moving around your data, you know you've done it correctly. And then you would just be able to paste that right into your Word document. Same with this one. You would just click on it, do Control C, and then you can paste that right into your Word document. I'm going to try to do that right now. I'm going to have to stop sharing this. And I'm going to have to share my Word document. Sorry, it's just going to take a second for me to get to it. Sorry, it's a lot of steps. Okay, here we go. Okay, here's my graph in my lab report. So I just did Control V to type. So Control C is copy, Control V would paste it in, and I could do the same thing with my data table. Okay, that's it. I hope your lab goes well. Thanks for your attention.